Okay, let's unpack this. Have you ever felt that incredible, almost cloud-like softness of alpaca fiber?
Oh, absolutely. It's really something else.
It's right up there with cashmere mohair, isn't it? Just that amazing feel, incredible warmth, and it holds up surprisingly well.
Yeah, it's durable too.
What's really fascinating about it, and kind of key to how it performs, is its structure. It has this unique air-filled channel right down the middle.
Like the medulla.
Exactly. And that trapped air makes it, well, significantly lighter and warmer than fibers like sheep wool. Some sources say, what, maybe 7 times warmer?
That unique structure definitely contributes to the luxury feel and the performance, yeah. But beyond just warmth and lightness, alpaca also has this beautiful luster.
Ah, yes.
It hangs really well, great drape, resists pilling, and generally it just takes up dye fantastically. It's a truly premium fiber, lots of good qualities.
It really is. And, while it takes dye well, the actual process of dyeing it, especially thinking industrially, well, it can be complex. And often it involves chemicals that aren't, let's say, ideal for the environment. And as you know, there's this huge growing demand for more sustainable practices everywhere, especially textiles.
Absolutely. The pressure is really on to find greener alternatives to those traditional processes. And this is where, chemistry and textile science are constantly trying to innovate.
Looking for ways to get the same results, maybe even better results, but with less environmental impact.
Exactly. Less impact.
Which brings us nicely to the source material for this deep dive. We're looking at a pretty recent study. It was published in Fibers and Polymers titled Dyeing of Alpaca Knitted Fabric Using Liposomes.
Ah, yes. This paper looks into, a novel approach to dyeing alpaca using something called liposomes as an auxiliary agent. A helper, basically.
Okay, so our mission today is to really dive into this study. We need to figure out what these liposomes actually are in this context.
Right.
Understand why the researchers thought, hey, maybe these could be useful for dyeing. See how they actually performed when dyeing alpaca fabric, especially compared to the more traditional methods?
Yeah, the benchmarks.
And figure out what the key findings tell us about maybe the future of sustainable dyeing. Does it hold promise?
I think it's a great example of how fundamental science, understanding the chemistry, the biology of materials right down at the microscopic level can directly impact the products we use.
The stuff we use every day.
Yeah, potentially making them better for us and for the planet. You'll see how this kind of cutting-edge research connects directly to that, that sweater or scarf you might be wearing.
Okay, let's jump into the study itself then. So the researchers, they focused on dyeing 100% alpaca knitted fabric. Simple enough. And they used a common reactive dye, Lanisol Red 5B.
Standard choice for protein fibers.
Right. And they set up experiments to compare 3 different dyeing methods. One used, no auxiliary chemical at all, just dye and fiber.
A control, basically.
One used a conventional anionic leveling agent, tested at 1% concentration.
The traditional approach.
And the third used these liposomes. They tested various concentrations, but for a lot of the direct comparisons, they focused on the 1% liposome concentration too.
Makes sense for a fair comparison.
Yeah.
And the core of this research really revolves around the liposome itself.
So what are they exactly?
Liposomes are microscopic vesicles. Think of them as tiny spheres made of lipid bilayers.
Lipid bilayers, like fats.
Yeah, essentially tiny bubbles with walls made of fat-like molecules. Very similar, actually, to the structure of our own cell membranes. They have a watery core inside.
Okay.
And they can trap substances like dye molecules either inside that watery core if the dye is water-loving, or within the lipid walls themselves if the dye prefers oily environments.
Huh. So they can hold different types of things.
Precisely. And they're already used quite a bit in fields like cosmetics, delivering active ingredients, and the food industry too. The idea here is leveraging them in textiles, maybe to deliver dyes or finishing agents more efficiently.
Okay, so they're like tiny fat-based delivery bubbles designed to carry the dye. And the study specifically wanted to see if these liposomes could work as an alternative to that traditional leveling agent.
Exactly. See, in traditional dyeing, particularly with protein fibers like wool or alpaca, especially under acidic conditions.
Which they used here, right? pH 4.5.
Yes, pH 4.5 for this reactive dye.
Sure.
Under those conditions, the dye molecules can sometimes just rush onto the fiber way too quickly.
Okay.
And this can lead to uneven color, blotchy dyeing, what the industry calls poor levelness.
Right, you want it smooth and even.
Exactly. And leveling agents are used precisely to slow down that initial, very rapid dye uptake. It gives the dye more time to distribute evenly across the whole fiber surface before it permanently bonds.
Okay, and how do traditional ones do that? The paper mentioned anionic leveling agents.
Right, so anionic just means they carry a negative charge. In those acidic conditions, the alpaca fiber develops positive charges on its surface.
Okay, opposites attract.
Exactly. So you have the negatively charged dye molecules wanting to bind to those positive sites. But the anionic leveling agent, also negatively charged, essentially competes with the dye for those same positive sites.
Ah, a competition.
Yeah. And the leveling agents tend to bind first, often because they're smaller, more mobile than the bigger dye molecules. They get there faster.
So they block the sites temporarily?
That's the idea. Then, over time, as the dyeing process continues, the dye molecules, which usually have a stronger affinity for the fiber, gradually kick out or replace the leveling agent molecules. This forces a slower, more controlled, and hopefully more lever saturation of the fiber with dye.
Got it. A controlled release, essentially, by initial blocking. But you mentioned a potential downside earlier.
Well, yeah, sometimes a small amount of that leveling agent might remain bound to the fiber even at the end of the process. It doesn't get fully replaced.
Okay.
And if that happens, it can slightly reduce the total number of sites available for the dye. The result might be a slightly lower final dye uptake.
Meaning a potentially lighter color than you could theoretically achieve.
Correct. A slightly lighter shade.
Okay. So that's the benchmark. How did the liposomes compare? The researchers looked at dye absorption over time, right?
They did. They tracked the exhaustion curves. And when they used the conventional anionic leveling agent, just as you'd expect, the dye uptake was significantly slowed down.
Especially at the beginning.
Especially early on, yeah. Which is exactly what you want for good levelness.
Mm-hmm.
But, confirming the potential downside, the final amount of dye absorbed at the end of the process was slightly lower than when they used no auxiliary at all.
Okay. Follows the theory. Now, the liposomes.
This is where it gets really interesting. When they used the 1% liposome concentration, the initial dye uptake was also retarded. It was slowed down compared to dyeing without any additives.
So check one, it does act like a leveling agent early on.
It shows that leveling effect, yes. But, and here's the twist, towards the end of the dyeing process, the samples dyed with liposomes actually showed higher final dye uptake.
Higher. Higher than both the no auxiliary and the traditional leveling agent.
Yes, higher than both.
Wow. So it helps achieve a level color and potentially gives you a richer, darker shade in the end. That sounds, well, like the best of both worlds.
It certainly seems that way based on these results.
Yeah.
The paper showed that the samples dyed with 1% liposome had a higher color yield. They measured this as the Ks value.
Ks value, higher means more dye.
Higher Ks means more dye absorbed on the fiber, yes. Okay. And they also had a lower L value compared to the other methods tested at the same dye concentration.
And lower L means darker color, right? In the C-Lab system.
Exactly. Lower L is darker. So darker color, more dye uptake overall.
But that feels a bit counterintuitive. How can it slow down the dye at the start and then help more dye get absorbed later?
Yeah, it's a good question. The researchers propose a couple of possible mechanisms, drawing on what we know about how liposomes and fibers might interact.
Okay.
The initial slowing down is likely pretty straightforward. The liposomes physically encapsulate or trap the dye molecules, right?
Mm-hmm. Like we said, the delivery caps.
Exactly. So that physically slows down their movement and diffusion towards the fiber surface. That explains the initial leveling effect.
Makes sense.
But the reason for the increased final uptake, that's where the hypothesis gets more interesting. They suggest that the liposomes themselves, being based on phospholipids.
Those fat-like molecules again.
Right. They could potentially interact with the fiber structure itself, maybe slightly loosening or altering the cell membrane complex within the alpaca fiber.
Ah, so interacting with the fiber's own sort of fatty components.
Potentially. And if that happens, it might make it easier for more dye molecules to penetrate deeper into the fiber, especially later in the process when temperatures are higher. This could allow for greater overall uptake compared to the other methods.
Okay, so the liposome acts like a temporary cage for the dye early on, controlling the delivery for levelness.
Mm-hmm.
But then it might actually interact with the fiber to make it like more receptive or porous to the dye later in the game. That's a really fascinating proposed interaction.
It is. And the statistical analysis in the study, the variance analysis, it confirmed that both the type of auxiliary used, none, traditional or liposome, and the dyeing time has statistically significant effects on that color yield, the Ks value.
So the differences were real, not just random chance.
Correct. Now, the statistical groupings did show some variations depending on the specific liposome concentration, for instance. They found that using 4% liposome actually resulted in a lower overall yield, more similar to the traditional leveling agent.
Oh, interesting. So more isn't necessarily better there.
Seems not. But the comparison at the 1% level, especially looking at those time-dependent uptake curves, clearly showed the potential for achieving both level dyeing and those darker, richer results with the liposomes.
What about the actual levelness, though? Did they measure how uniform the color was? Because that was the original point of a leveling agent.
They did, yes. They calculated levelness percentages based on measurements across the fabric surface in their lab setup. They found the conventional leveling agent gave the highest levelness score at 98.0%. The liposome method came in very, very close behind at 97.6%.
That's barely a difference.
Very close. And dyeing without any auxiliary was, as expected, lower at 96.7%. So the liposome performed almost identically to the traditional leveling agent in terms of uniformity, at least under their specific lab conditions.
While giving that higher final dye uptake.
Exactly. And they do correctly point out that really assessing levelness properly needs bigger trials. Industrial scale dyeing is different.
Sure, lab conditions are one thing, a giant vat is another.
Okay, and the color itself, apart from being darker with the liposomes, did the actual red shade change? Was it a different kind of red?
Good question. They measured the full Scilab values to get a more precise description of the color. So, as we said, the L values confirmed the liposome sample was darkest, lowest L.
Right.
Then they looked at the a value positive A is redder, negative is greener, and the B value positive is yellower, negative is bluer.
Compared to dyeing without any auxiliary, the liposome dyeing resulted in a slightly more red nuance, so a higher value,
A bit more intense red.
and a slightly less blue nuance, meaning a higher B value, shifted slightly towards yellow. The traditional anionic leveling agent sample, conversely, was slightly less red and slightly more blue.
Subtle shifts. What about vibrancy?
Ah, yeah, chroma or C. That tells you how vibrant or saturated the color is. The liposome dyeing method produced the most saturated color, the highest C value,
Okay.
and the traditional leveling agent dyeing was the least saturated. Interestingly, the hue angle, HQEs, which represents the overall basic shade type, didn't show a statistically significant difference between any of the methods.
So summing that up, with liposomes, you get a level color, it's darker, it's slightly more saturated or vibrant, and there's a very subtle shift in the red, blue, yellow balance compared to the other methods.
That's a good summary, yes.
Okay, that all sounds very positive.
Yeah.
Level, darker, more saturated. But that leads to the really critical question, right? The practical stuff, like fastness.
Absolutely crucial.
You don't want that beautiful dark color just washing out in the first wash or rubbing off on your sofa.
No, definitely not. Any new dyeing method has to pass those tests to be viable. So the study did test wash fastness, dry rubbing fastness, wet rubbing fastness, and also light fastness. They used standard ISO textile testing methods.
And the results?
The results were good news. Basically, all the samples, whether they used no auxiliary, the conventional leveling agent, or the liposome, showed very good fastness properties across the board.
No significant difference between them.
The ratings were comparable for all the methods tested, which is a really significant positive finding. It means using the liposomes didn't compromise these essential practical properties.
That's fantastic. Okay, so let's try and boil down the core findings reported in this study then.
Well, I'd say the main takeaway is that these liposomes, they can effectively function as a leveling agent during the dyeing of alpaca. They help achieve that uniform color by slowing down the initial dye rush.
Right, the primary goal.
But, critically, they seem to offer an additional benefit. They actually promoted higher final dye uptake compared to the traditional methods tested in the study.
Which results in a richer, darker color.
Exactly, and all this without negatively impacting the crucial fastness properties. Wash, rubbing, light fastness were all good.
Okay. And why does this matter in the bigger picture? What's the significance here beyond just getting a really nice, dark, even red on some alpaca fabric in a lab?
Well, it's potentially highly significant, especially in the context of the textile industry's big push towards sustainability.
Right, the green chemistry angle.
Exactly. The study itself highlights that using sustainable chemicals, like liposomes could be, is gaining real importance. Because liposomes are typically derived from natural sources, phospholipids mainly.
From plants, like soy, or even animal sources.
Often soy, yeah. They're generally considered more eco-friendly auxiliaries compared to some of the synthetic chemicals traditionally used in dyeing processes. Finding effective, sustainable alternatives like this one could be. Well, it's increasingly important if we want to make the textile industry greener.
It certainly is. This research really feels like maybe a solid step forward in finding more environmentally conscious ways to process these beautiful, luxurious natural fibers like alpaca.
It's promising, definitely.
Now, the study itself, it concludes, as many studies do, by suggesting, hey, it would be really valuable to test these liposomes under proper industrial scale conditions.
The scale-up question.
Yeah, to really compare their technical feasibility, their economic viability, how much does this cost, and the overall ecological performance against the existing conventional leveling agents in a real factory setting.
And scaling up any process, moving it from a small lab beaker to a massive factory dye machine, that always presents unique challenges. It's never straightforward.
No, absolutely. And that kind of leaves us with a thought for you, the listener, to maybe mull over. The study points directly to the need for these industrial trials. So what challenges or maybe what opportunities do you think might arise when trying to take this liposome dyeing process from the, relatively controlled environment of a lab and actually implement it on a large factory scale?
Yeah, what happens in the real world?
What might the economic hurdles be? Are liposomes actually cost-effective to produce and use in the huge bulk quantities needed compared to the existing chemicals, which are often quite cheap?
Good point. Cost is always king.
And what about the technical challenges? In a huge dye vat versus a small lab machine, things like ensuring you get consistent liposome dispersion throughout thousands of liters of liquid, or managing process parameters like temperature ramps, flow rates on that much larger scale. Does it behave the same way?
Consistency is key at scale.
And then, thinking even broader, what are the ecological impacts to consider at that large factory scale, beyond just swapping one chemical for another? Things like overall water usage, energy consumption for the process. How do you handle the waste streams? Does using liposomes change any of that calculus significantly?
Thinking about those real-world implementation factors, the whole life cycle, that's really key to understanding the full potential of this kind of innovation, and whether it can genuinely shift the industry towards being greener.
Absolutely. It's one thing for it to work in principle, another for it to work in practice, economically and ecologically.
Well said.
Well, thank you for joining us for this deep dive into what is, I think, a really fascinating corner of fiber science and sustainable chemistry.